Thank you very much for the introduction. Excited to be here presenting to you my company. We are a spin-out out of MIT, specifically from the mechanical engineering department, my co-founder's lab, and wanted to demonstrate to you that there is now a platform that truly scales between the research and manufacturing pipelines. What we're trying to do is we're really trying to improve patients' lives using automated cell engineering. The technology that we have developed is called FlowFect, and this can be implemented in low volumes, so high throughput discovery, and without having to re-optimize or change equipment, we can go into a continuous flow manner for cost-effective cell therapy manufacturing. The traditional paradigm in discovery is really to have a platform that you can enable many different parameters in a high throughput way that is then put through a process in order to scale up towards a therapeutic potential. And this is being done with viruses. Many of the early discoveries in the cell and gene therapy were done with viral delivery, and the importance here is that once you have a discovery in the lab, then there is a time frame that is significant to be able to get your time slot and scale up for manufacturing. This also carries a heavy cost associated with it that is not negligible for small biotech companies. There's non-viral solutions in the market, and some companies are using a particular technology for discovery and then trying to transition into a different platform for their GMP manufacturing. And again, these are different companies or not give you the ability to really optimize your parameters seamlessly. What I'm here to tell you today is that we have developed a platform that eliminates the need for a scale-up. And this is what we call FlowFect. It's a continuous flow-through technology in order to deliver genetic material to a wide array of cells and a wide array of payloads. We have a high-throughput platform, which we call the FlowFect Array. And in this platform, you can process very small volumes, volumes on the order of 25 microliters to 200 microliters. And the two applications that you can use this platform for is to optimize critical conditions of your delivery or to conduct CRISPR screen type libraries. Without changing the platform, we can go into a continuous FlowFect TX that employs a cartridge-based system. But the important thing is that the technology that is fundamental to both applications does not change. What you can do in a pipette tip, you can also do in a cartridge-type device. So I'll walk you through a video on which shows how this actually works. Here we have a commercially available liquid handling system. We have optimized our pipette tip devices with the electrodes in order to integrate into the system. We have these devices aspirate your payload and your cells that are already pre-mixed in this 96-well format. We introduced some air gap to put the sample above the electrodes. And the important thing is when we're dispensing the sample out of these pipette tips, that tips are energized and exposed to just the right amount of electrical energy to deliver efficiently and maintain high cell viability. The flow rates are high. The device geometry is actually large as well, and this means that the process is extremely gentle to the cells while maintaining high cell delivery efficiency. We have now the ability to program 96 independent combinations of electric fields and flow rates in order to really optimize this delivery in less than 10 minutes. Once you optimize, you can envision going into different genetic payloads for each well and identifying therapeutic targets in a rapid manner. Some of the data that we have generated is demonstrated here on the screen. These are jerkets. It's a cell line for lymphocytes. Viabilities of these cells are extremely high, very comparable to non-treated samples. We have delivered DNA and mRNA. And the experiment here was really comparing what happens if I do 1 pipette tip at a time or if I do 8 in parallel. The results demonstrate that both are possible, and we are super excited about that because that really means that you can increase the throughput and continue to build capabilities so that we can envision a 96 or 384 type of format, because liquid handling systems already exist that can manage this throughput. But we have not just been working with cell lines. The data that I'm going to present right now is actually flow cytometry data in primary T cells. These are healthy donors, day 10. And for those of you that are not familiar with flow cytometry, on the top what we have is our total cell gate for non-treated samples. On the bottom, we have the cell viability gate. Non-treated samples, viability is on the order of 81%. When we process these cells with commercially available electroporation technologies, what we have seen is that there is a shift in the morphology of these cells at 24 hours with a big hit in terms of viability of these cells. This is reducing to on the order of 30% viability. And remember, right here, we are really gating on the total cells that you put in, not just focusing on that lymphocyte bubble. Our technology has been able to maintain the phenotypic relationship of these cells. You could virtually superimpose the cells that are treated with flow effect with the non-treated, and our viability remains unaffected on the order of 84%. So some of the studies that we have ongoing are demonstrating different payloads, different cell types, and always maintaining this high performance yield that is competitive in the market. I mentioned that we have 2 platforms. The second platform is the FlowFect TX system, in which the cell is now flowing in a tubular structure. So the flow is driven by a peristaltic pump as opposed to a liquid handling system. But the flow rates, the electric fields, the buffers are identical. Here, this demonstration is showing that it can be done in a continuous flow manner. There is no need to change the geometry of the cartridge. The channel is the same. And you're continuously flowing through the area of high electric field. These flow rates are actually very important for us. They're on the order of tens of milliliters per minute, which means that at a cell concentration of 100 million cells per ml, you can process cells at the rate of 1 billion cells per minute. Data that we can show publicly here demonstrates our ability to process 100 million T cells in 30 seconds, 15 seconds, or 3 seconds. And the way this data was collected was to try and process at 3 different cell concentrations. The curve or the bar on the left is a cell concentration of 10 million cells per ml, and this is a volume of 10 mLs. The bar all the way on the right is a cell concentration of 50 million cells per ml, so that is a 2 ml volume. What's important here is that if we're processing 10 mLs or 2 mLs, we are getting consistent yield, which is the live number of cells that are expressing the protein of interest from the input number of cells. So we put in 100 million primary T cells, and the result is on the order of 60 million that are alive and expressing the GFP protein in this case. Really important is we reduced the amount of mRNA that we used in this experiment by a factor of 5 because we kept that ratio of mRNA to sample constant to 10% of that volume. And we can see that we can reduce that amount without compromising the yield, which is really an impact on potential cost of goods sold savings for our partners. So, in summary, our technology can process 96 well plate, 96 conditions within minutes. With our other technology, we can process cells on the order of 1 billion cells per minute, but the flow rates and the electric fields from one platform translate directly into the other platform. That's really that seamless scalability towards the clinic. The cell health is maintained because the flow rates are high, which means that the cells only experience that electric field for orders of seconds, and then they go directly into the recovery media, which has demonstrated some tremendous promise. We have processed multiple payloads, DNA, mRNA, CRISPR, Cas9, RNP, including delivery into the nucleus, and some of the cells that we have tested are primary monocytes, primary macrophages, primary T cells, including naive T cells as well, with very high yields. Right now, we have several active pilot studies ongoing. The strategy is really to be able to demonstrate the power of the technology with our partners' own cells on our partners' own payloads. And the programs that we are pursuing right now are to engineer CAR-Ts, engineer TCRs, and also to knock out endogenous genes using CRISPR technology. We see several opportunities in this conference alone, and really what we want to do is accelerate the development and discovery of next generation cell therapies. We want to be able to identify those clinical users that are willing to do alpha testing for our prototype, which will become available in the next 6 months. And we want to expand beyond our primary T-cell studies that we have ongoing and expand with partners that have expertise in other cell types, such as stem cells, natural killer cells, macrophages, and patient cells. At the end of the day, we want to help you think about the next generation therapies and transition some of your viral programs to non-viral programs if there's opportunity to engage. Our team is composed of scientists with expertise in adoptive cell therapy, CRISPR delivery, engineers in terms of automation and R&D, and really expertise in microfluidics coming from Colin's lab, my co-founder, who is in the audience as well. Recently, we've received several different awards. We are in a stage that there is a real need in the industry, and we are trying to address that need in order to enable all of you in the audience that may have some of these challenges. So with this, I would like to thank you for your attention, and I'm happy to take any questions if there is time.